Welcome back to Catholic Courses and the 7 Threats Against the Culture of Life. I'm your professor, Dr. Scott Gaylord. In this lecture, we will discuss what, at one point, was thought to be the stuff of science fiction, human cloning. Have you ever considered what it would be like to have a clone? A genetic duplicate that could help you get through another busy day. Children sometimes dream about sending a clone to school or having a clone do their homework. Adults might use their clones to do errands or those less desirable chores like laundry, cutting the grass, or painting. And in the movies, such as Star Wars, The Clone Wars, genetic copies can fight our battle so that real people do not have to get hurt. Or perhaps when you hear about cloning, you think about Jurassic Park, using preserved DNA to bring back extinct species such as the dinosaurs. Cloning makes for interesting thought experiments and oftentimes entertaining movies. But what, or perhaps more appropriately, who is a clone? In our daydreams, clones are more like look-alike robots who can pass for us, but who are not us. They are automatons that can be used for our pleasure and then turned off or even discarded when we are done with them. In the real world, though, clones are living, breathing organisms. In the case of Dolly, the first cloned mammal, it was a living, breathing sheep. If a person is cloned, there is a living, breathing person. But if human clones are persons, then they are subject to the moral law, and our actions toward them also are either moral or immoral. As a result, in this lecture, we will evaluate the science and ethical considerations surrounding the creation and possible use of clones. The propriety of scientific cloning reminds me of something I sometimes tell my children. Just because you can do something doesn't mean you should. Pope Benedict XVI put it much more eloquently in his address to a stem cell conference in 2006. Progress becomes true progress only if it serves the human person and if the human person grows, not only in terms of his or her technical power, but also in his or her moral awareness. Contrary to some critics, the Catholic Church is not hostile to science. Rather, as Pope John Paul II stated, the Church is convinced of the need to support and promote scientific research for the benefit of humanity, provided that such research is respectful of the dignity of the human being. Both of these popes recognize that scientific inquiry is critically important, but that scientific advances must be pursued in an ethical manner, and not simply because they are technically possible. Yet human cloning threatens to fall into this latter category. Having figured out how to perform in vitro fertilization and that embryonic and adult stem cells can be manipulated to develop into specific types of tissue, scientists began considering whether they could clone animals. And once science cloned the first mammal, some researchers sought to clone humans as well, viewing human cloning as a possible way to help infertile couples as well as those suffering from various diseases. Although cloning frequently is associated with science fiction, a type of cloning occurs naturally in some pregnancies. Identical twins are clones of one another, unique individuals that happen to be genetically identical. From a purely technical standpoint, then, cloning involves the creation of a genetic copy of a particular DNA sequence or the entire genome of an organism, as occurs with human twins. Unlike identical twins that occur normally in roughly 0.3% of the births, artificial cloning is an asexual or agametic reproduction process. That is, there is no sexual union of man and woman, or even sperm and egg as in IVF. Instead, the clones are created in vitro, in a glass container or petri dish in a lab. There are generally 2 forms of cloning. The first is called artificial embryo twinning, which consists in the artificial separation of individual cells or groups of cells from the embryo in the earliest stage of development. The separation process mimics the natural splitting that occurs in utero when twins are formed naturally. The separated cells are allowed to develop independently and then are transferred into the woman's uterus where, if the pregnancy is successful, they develop as identical twins. The second type of cloning, and the one that causes the most public debate, involves a particular scientific procedure known as somatic nuclear cell transfer, or SCNT. In SCNT, the nucleus of a somatic cell, which refers to any cell other than a sperm or egg cell, is transferred into an oocyte, an unfertilized egg cell that has had its nucleus, and therefore most of its DNA, removed. Because the nucleus of the somatic cell already contains a complete DNA and chromosome structure, SCNT does away with the need for a sperm to fertilize the egg. Once the nucleus is inserted into the egg cell, the newly constituted oocyte is then stimulated with chemicals or electricity to cause cell division, which helps the embryo grow. Because the nuclear DNA of the newly created embryo is from a somatic cell of the donor, the embryo and the donor are, for the most part, genetically identical. I say for the most part because SCNT typically does not result in an exact genetic duplicate of the donor of the nuclear DNA. As it turns out, genetic material actually comes from two different sources. First, the nucleus of the donated somatic cell, and to get even a little bit more technical for just for a minute, second, the mitochondria contained in the cytoplasm of the oocyte. That is, a part of the egg that is outside the nucleus. Mitochondria provide power for the cell and also contain short DNA segments. Thus, unless the egg came from the somatic cell donor, the clone will not be the exact genetic twin of the egg donor. Take, for example, Dolly, the first mammal to be cloned using SCNT. In 1997, a team of scientists at the Roslin Institute in Scotland took the nucleus of a cell from the mammary gland of a Finn Dorset sheep and inserted it into a denucleated cell from a black-face ewe. The newly created embryo was then placed in a surrogate ewe, and roughly 5 months later, Dolly was born, sharing the exact same nuclear DNA as the Finn Dorset sheep, from which the somatic cell was taken. Since 1997, animal cloning has become much more widespread. In fact, in 2008, the United States Food and Drug Administration approved the consumption of meat and milk from cloned animals. As SCNT improves and allows for the efficient production of cloned animals for consumption, we likely will see more and more cloned animal products for sale in the marketplace. At the time, though, when Dolly was born, her birth was heralded not simply because it showed that cloning mammals was possible, but also because it demonstrated that differentiated cells, cells that had developed specific functions, like the mammary gland from which the somatic cell for Dolly was taken, could be reprogrammed and effectively act like a newly fertilized egg. That is, SCNT enabled scientists to cause differentiated cells to revert back to an earlier undifferentiated state, creating a completely new, albeit genetically identical, organism. The process, though, was not as easy as this quick summary may make it seem. SCNT puts tremendous stress on both the somatic cell nucleus as well as the denucleated egg cell. In fact, Dolly's birth occurred only after 277 eggs underwent the SCNT process. That led to 29 viable embryos, but only one Dolly. Think about that for a second. The success rate on Dolly was roughly 0.36%. One live birth for the 277 embryos created. Now imagine if that process was used to create human embryos. Regrettably, this is not idle speculation. In 2001, scientists in Massachusetts cloned several human embryos, but only one developed into a blastocyst, roughly 32 cells, at which point cell division stopped. To date, there have been only a limited number of cloned human embryos. From these human embryos, at most one stem cell line originating from Korea has survived. Yet to create this one cloned human stem cell line, at least 166 cloned human embryos were destroyed. Consequently, cloning of human embryos, like embryonic stem cell research generally, has not led to a single successful treatment or cure.